Hello, 8th standard Cbse children. Have a good day. Have a nice day. This is our video number 18. We are continuing the lesson chapter 2 microorganisms friend and foe. Friend we are dealing with useful activities and foe means dealing with harmful activities. I hope you understand. So this first part we have learnt about agents of microbes, infectious agents of microbes. Then we learnt about viruses, bacteria, fungi. Now we are learning about useful and harmful activities and also a very little information about food preservation. Right? Already we have learnt Anton von Leeuwenhoek was one of the first persons to observe microorganisms using microbes of his own design. Leeuwenhoek is commonly known as father of microscopy and microbiology. And then we have learnt as bacterial structure, fungus with the body. The body of fungus is formed of mycelium, which in turn formed of numerous filaments called hyphae. I hope you remember all that. And then Louis Pasteur discovered fermentation in the year 1857. Yeah, and the useful organism, as we have learnt, as food and beverage industry, they are useful in food and beverage industry, medicinal use, increase the soil fertility. We are going to learn about nitrogen cycle and maintain clean environment. What is given in our textbook? They are food and beverage industry, medicinal use, increase soil fertility, maintain clean environment. But this food and beverage industry, we are learning about curd is formed by coagulation of a protein called casein, which is present in milk. As in spelling CASEIN. And bacteria lactobacillus promotes curd formation by converting lactose sugar in milk into lactic acid. It multiplies in warm milk and converts it into curd. About that? Then cheese and paneer. The production of milk products such as cheese and paneer involves the use of bacteria, lactobacillus and streptococcus. Streptococcus fungi. Okay. And some fungi like aspergillus and penicillium are also added to cheese to give a special flavor. The figure aspergillus and penicillium, but video 17 the first picture, that is aspergillus and penicillium, right? And then fermentation process in bread and idli dosa. I hope you understand. Can you? கொஞ்சம் recollect பண்ணு. Can you? Next, food industry, alcoholic beverages, vinegar, citric acid and sources of nutrients. Algae, some algae like chlorella and seaweed are used as food in many countries. Chlorella is very rich in proteins and vitamins. Seaweeds are rich source of minerals and all natural vitamins. I hope you understand. So this is video number 18. Whereas harmful activity, 4 children. In detail in the video I am just giving a brief introduction. Right? Some of the common communicable diseases in human being, and along with their mode of transmission, causative, pathogens and preventive measures we are going to learn. Tuberculosis, cholera, typhoid, poliomyelitis, measles, chickenpox, common cold, hepatitis and malaria. They are common diseases in man. I repeat, tuberculosis, cholera, typhoid, poliomyelitis, measles, chickenpox, common cold and hepatitis. As we know, cholera, waterborne disease, tuberculosis, airborne disease, typhoid, again waterborne, polio, water and airborne disease, measles, airborne disease, chickenpox, airborne disease and any physical contact, common cold, airborne disease, hepatitis A is again airborne diseases. I hope you understand. And then animal diseases, foot and mouth diseases, anthrax and fin rot. It is given fin rot. Otherwise, we can also say rust of wheat and blast of rice. Rust of sugarcane. They are also plant diseases. Right? And citrus canker, yellow vein mosaic of bindi, lady's finger. They are also as plant diseases. So these are the plant diseases, and again I repeat, in video number 18 we are learning as second part of the lesson in our textbook. So we'll continue this video session. Video 18, chapter 2, microorganisms, friend and foe, part 2. I hope you understand. So we'll just continue. Hello, 7th, 8th standard. Have a good day. Have a nice day. Today, this is our 8th video session where we are going to do the lesson Microorganisms, chapter 2 in our textbook as friend and foe. Right? Now this microorganism, friend and foe, we are going to do part 2. Part 2 already in video 7 we have learnt the first half of the lesson as what are microbes, as those infectious agent or microscopic agent, and then usefulness of microbe. We have learnt as usefulness of microbes. That I will just give a gist, where we learnt about Anton von Leeuwenhoek, who was the first scientist who studied and discovered the microscope, and he studied the structure of bacteria under microscope. Right? So microscope, it is an instrument that is used to see small objects, minute objects, tiny objects that are not visible to the naked eye. Okay, now these microorganisms are also called microbes that can be seen only with the help of microscope. And it was Louis Pasteur who did fermentation process, that is the process of conversion of sugar into an acid or alcohol by the action of microbes. Alcohol, now here it is ethyl alcohol, and acid is with reference to lactic acid. As I said earlier, last year anaerobic respiration படிச்சோம், at accumulation of lactic acid in the muscle cell resulting in muscle cramp, and the lactic acid formation. Then pathogens are harmful organisms. Pathogens are harmful organisms which cause disease in human being, plants and animals. I repeat, pathogens are harmful microbes which cause diseases in human, plants and animals. Then we learnt about communicable and non-communicable diseases. Otherwise, infectious and non-infectious diseases. Communicable diseases that can spread from an infected person to a healthy person through air, water, food or physical contact. Same way, non-communicable, it will not spread to others. It is formed within the body due to malfunctioning of any of the system. Example, diabetes. And then we are going to learn, now we are going to learn about harmful activities of microbes and this food preservation method to prevent all this harmful activity and to make use of the usefulness of microbe, how the food should be preserved. That is also a part of the lesson. Now in usefulness of microbes நம்ம நிறைய படிச்சிருக்கோம். As usefulness in food industry, and then usefulness of algae, and then agriculture, uses of usefulness of microbes in agriculture as root nodules of leguminous plants படிச்சோம்ல, அந்த இதெல்லாம் as friendly microbe. I hope you are following. Then now next we are going to do harmfulness of microorganism. Microorganisms are harmful in many ways. Some of the microbes cause diseases in human, plants and animals. Such disease causing microorganisms are called pathogens. Some microbes spoil food, clothing and leather. Food தெரியும் ஸ்பாயில் ஆயிடும். லெதர் நம்ம அந்த மழை காலத்தில் இந்த லெதர் செப்பெல்லாம் அப்படியே போட்டிருந்தோம்னா அந்த பூஞ்சை பிடிச்சிருக்கு சொல்லும்ல, இந்த பஞ்சை அந்த லேயரை பேட்சி ஒயிட் பேட்சஸாக இருக்கும், அதுதான் லெதரை இட் இஸ் அட்டாக்கிங். அண்ட் ஆல்சோ க்ளோதிங். சரியா? So now the other one, this microorganism, it is briefed about advantages and disadvantages that you could go through. சரியா? நான் படிக்க வேண்டாம், that itself is self-explanatory. அதில் மெயினாக டிசீஸ் காசிங் மைக்ரோ ஆர்கானிசம், நம்ம மெயினாக வைரஸ் படித்தோம்ல, viruses which attack bacteria, they are called bacteriophage. Which attack plants, phytophage. Which attacks animal, zoophage. Algae, phycophage. And fungi, mycophage. இல்லையா? இது viral attack. இதே மாதிரி plants and animals are also attacked by these harmful microbes. I hope you are able to follow this. So now the next one is about harmful microorganisms. Microorganisms are harmful in many ways. Some of the microbes cause diseases in, இப்போ அதே தான் ரிப்பீட் ஆயிருக்கு, plants, animals and human. Such disease causing microbes are called pathogens and they spoil food, clothing and leather. So now we will see the harmful activities of bacteria. சரியா? Sorry, harmful activities of microbes. அதில் மெயினாக இந்த பிக்சர் பாரு, இது ஒரு டேபிள். I hope you could see clearly. இது ஒரு டேபிள். இது how it is caused by bacteria, viruses and fungi னு ஒரு டேபிள் கொடுத்திருக்கு. நான் அந்த கியூபில் தரேன், அதை நீ கொஞ்சம் டீட்டெயில்டாக பார்த்துக்கோ. சரியா? So now the diseases that spread from one person to another is called infectious diseases or communicable diseases. நாம் அதை ஏற்கனவே ஒரு தடவை படிச்சிருக்கோம். So I repeat, diseases that spread from one person to another are called infectious diseases or communicable diseases. Microbes can enter our body through air we breathe, the water we drink, cuts in the skin, and insect or animal bites. And some of them are leprosy. Leprosy is a contagious disease caused by bacterium. So, bacterial diseases are leprosy, tuberculosis, cholera, anthrax and tetanus. These are all bacterial diseases. I repeat, leprosy, tuberculosis, cholera, anthrax and tetanus. These are all bacterial disease where leprosy through air and direct contact, tuberculosis through air, cholera through water, anthrax through air, direct contact and infected food. நம்ம வந்து mode of transmission படிச்சோம் இல்லையா, as airborne, foodborne, waterborne and vector-borne diseases. அதான் இது. சரியா? Then amoebiasis caused by amoeba. It is a protozoan through infected food or water. Then malaria, again it is a protozoan disease caused by the protozoan Plasmodium and by the bite of female Anopheles mosquito. Then athlete's sleeping sickness, again it is protozoan disease caused through insect bite. Athlete's foot, இது வந்து இந்த சொறி படை சரங்கு சொல்லுவல அது, caused by fungus through infected clothing or places. Flu, chickenpox, measles, dengue and rabies, they are all viral diseases. I repeat, flu, chickenpox, measles, dengue and rabies, these are all viral diseases. சரியா? Where common cold, flu, chickenpox and measles, they are airborne diseases or through direct contact, and dengue fever caused by insect bite, and then Aedes aegypti சொல்லுவோம்ல mosquito, Aedes aegypti, and then rabies caused through bite of rabid animal, dog and cat, mainly dog நாய் கடிச்சு அந்த வெறி நாய் கடிச்சதுன்னா people will be suffering from the disease as dengue fever, rabies. சரியா? This is for viral diseases. What are all the viral diseases? Common cold, flu, chickenpox, measles, dengue and rabies. I hope you are able to follow this. So these are the mode of infection or method of infection. சரியா? இதுல anthrax is a lethal disease of cattle caused by bacterium. Anthrax and the lethal disease leading to death. Anthrax is a lethal disease of cattle caused by bacterium. In the Second World War, I hope you have heard about it. Second World War la என்ன பண்ணானா இந்த soldiers எல்லாம், the foreign soldiers வந்து என்ன பண்ணா, the anthrax ஓட spores, it is highly poisonous. இந்த anthrax ஓட spores அவளோட arrowல அந்த அம்பு விடுவாள் இந்த வாரும்போது சண்டையில் இந்த anthrax spores முக்கி அதை வந்து அம்பு எய்தினா அது வந்து that killed many of the enemies, and the other end level enemies, it killed many soldiers, and this is highly poisonous. So it is normally termed as biological weapon. War la use பண்ணனால spores of anthrax is called biological weapon. சரியா? Then avian tuberculosis is a bacterial disease of poultry. Poultryனா இந்த கோழி பண்ணைலாம் சொல்லுவோம்ல அந்த இது. Avian tuberculosis is a bacterial disease of poultry, and bovine viral diarrhea and foot and mouth disease are important viral diseases of cattle. I repeat, bovine viral diarrhea and foot and mouth disease are important viral diseases of cattle. And swine flu is caused by influenza virus in pigs. Swine flu is a disease caused by influenza virus in pig. Okay. Now this is a diagram caused by foot and mouth disease. I hope sorry. It's a diagram caused by foot and mouth disease. The கீழே இருக்கிற diagram. சரியா? So foot and mouth disease of cattle. அது வந்து வாயிலையும் புண்ணு வரும், கால்லையும் புண்ணு வரும். அது பேரு foot and mouth disease. சரியா? Then some kinds of algae produce toxins that can kill human fish and livestock as cattle, goat and so on. So this is a brief introduction for disease causing microbes. Okay, now there are some insects and animals as carriers. There are some insects and animals which act as carriers of disease causing microbe. Housefly is one such carrier. The housefly வந்து நார்மலாக அந்த கார்பேஜ் வீட்டு குப்பையிலலாம் இருக்கும் இல்லையா, அது அங்கெல்லாம் உட்கார்ந்துட்டு அது when it is visiting another ஃபுட்டில் வந்து அதில் ஏதாவது germ இருந்ததுன்னா that will cause food spoilage. சரியா? The flies sit on the garbage and animal excreta. Pathogens stick to their bodies. When these flies sit on uncovered food, they may transfer the pathogens. Whoever eats the contaminated food is likely to get sick. It is advisable to always keep the food covered. Avoid consuming uncovered food item. And another example, இது வந்து housefly. Another example, it is female anopheles mosquito which is causing malaria. Right? Malaria வந்து நார்மலா கொசு கடிச்சதுன்னா it will just enter into it, carries the pathogen called protozoan plasmodium. So in the malaria, when the mosquito when it bites female anophel, அதோட saliva வந்து இந்த pathogen இருக்கும். So that will enter the human body and that will be circulated within the RBC blood circulation for half an hour. And then it reaches the liver. There it will start multiplying. So malarial parasite வந்து ரெண்டு host, ஒன்று வந்து mosquito, another வந்து human. So mosquito is a primary host, human is a secondary host னு சொல்லுவா. I hope you understand. So example carrier is the female anophel mosquito which carries the parasite of malaria. Then female Aedes aegypti acts as a carrier for dengue fever, dengue virus. And all these mosquitoes breed in water. Hence, one should not let water collect anywhere in coolers, tires, flower pot, etc. By keeping the surrounding clean and dry, we can prevent mosquitoes from breeding. I hope you are able to follow this. So this is for vector-borne disease, how it is affecting. Okay. Now here, this is our book example. Some common plant diseases caused by microbes as citrus canker. It is a bacterial disease, Xanthomonas citri. It is Xanthomonas citri and it is airborne disease. The lemon, natai, the fruit dot brown dot, that is the citrus canker. Next one, the rust of wheat caused by fungi and mode of transmission air and seeds. Then third one, yellow vein mosaic of bindi. Bindi na lady's finger. Yellow vein mosaic of bindi, it is a viral disease caused by insect. So citrus canker, rust of wheat and yellow vein mosaic of bindi. These are the three textbook example of plants. Other few example I am giving. Crown gall disease in cotton is caused by bacterium. I repeat, crown gall disease in cotton is caused by bacterium. Citrus canker, then rice blast is caused by fungus. Then Panama wilt is a fungal disease that affects banana plant. Panama wilt is a fungal disease that is attacking the banana plant. Red rot of sugarcane, fungal disease. Rice tungro disease caused by tungro viruses. So other additional example that I am giving: crown gall, rice blast, Panama wilt, red rot of sugarcane and rice tungro diseases. These are also plant diseases attacking different types of plants. I hope you could follow this. And then as animal diseases, next what we are going to see is diseases in animal and human, tuberculosis, measles, chickenpox, polio. Again, cholera, typhoid, hepatitis B and malaria. These are all diseases attacking the human. So I repeat, tuberculosis as tuberculosis, measles, chickenpox, polio, cholera, typhoid. The tuberculosis, cholera and typhoid, they are bacterial diseases. Measles, chickenpox and polio, they are viral diseases. Hepatitis B is viral disease. Malaria is a protozoan disease. I hope you could follow this. And then mode of transmission this is how you have to learn. It is already given in the book, so that I am not reading. You have already learned this. I am just summing up this lesson. Okay. The next one is about food preservation. Food preservation. Food should be preserved due to food spoilage. Food products can become contaminated. I repeat, food products can become contaminated by bacteria and fungi, which can spoil milk, meat, fish, vegetable and fruit. So spoiled food contains toxins produced by the microbe, which can either reduce the taste or smell of the food, causing illness, and that is food poisoning. And the action of yeast on fruit juice makes it unsuitable to drink. This is because yeast converts sugar into the juice to alcohol, or sometimes wine, I know. And then the presence of some kinds of microbe in food can cause infectious disease. For example, Salmonella bacteria in meat and eggs cause the disease typhoid. Okay, so food preservation should be done properly. So now what is food preservation? It is the process of preservation of food from decay or spoilage by storing in a condition fit for future use. Food is preserved to increase the shelf life of food, retain color, texture, flavor and nutritive value, increase food supply and decrease wastage of food. What does it mean? Increase the shelf life of food. Suppose or 1 month food வாங்கி வச்சிருந்தீங்கனாக்க 1 month food when you are collecting, it should keep up the good condition of the food, that is shelf life. So it increases the shelf life of food, retain color, texture, flavor and nutritive value. So food preservation is done to increase the shelf life of food, retain color, texture, flavor and nutritive value, increase food supply and decrease wastage of food. So for these reasons, food is preserved. Retain the color theriyum, texture na and the consistency and the idum theriyum, flavor and the taste, aroma, smell cholluvom. It should not be disturbing all the nutritive value. So food preservation is done for that. Now methods of food preservation. நம்ம bookல சிலது இருக்கு, சிலது இல்லாததும் நான் இதில் சேர்த்திருக்கேன். Methods of food preservation. First one, drying, dehydration, pickling and salting, cold storage, freezing, pasteurization and canning. இந்த pictureல இது drying, கருவாடு காய்ச்சறால காயப்பட drying. இது pasteurization. Louis Pasteur identified the method of pasteurization by heating the milk into the given temperature and then cool. This is pasteurization. இது வந்து cold storage. நம்ம வந்து சூப்பர் மார்க்கெட்லலாம் பார்த்தோம்னா இந்த பெரிய ஃப்ரீசர் பாக்ஸுக்குள்ள இந்த ஸ்டோரேஜ் மெட்டீரியல் போட்டுப்பல ஃப்ரோசன் ஃபுட், தட் இஸ் கோல்ட் ஸ்டோரேஜ். அண்ட் ஃப்ரீசிங், அதுவும் ஃப்ரீசிங் மெத்தட் தான், இந்த ஃப்ரீசிங் மெத்தட். அண்ட் தென் பாஸ்டரைசேஷன் இது. தென் கேனிங்னா இந்த டப்பாவில் அடைச்சி வச்சுருப்பால டின்னில், அதுதான் canning. So methods of food preservation: drying, dehydration, pickling and salting, cold storage, freezing, pasteurization, canning. Right? Now here, for this preservation of food, there are two methods of preserving by adding some preservatives. One is natural preservative, another one is artificial or synthetic preservatives. Natural preservatives are addition of salt, sugar and oil. Addition of salt or sugar or oil. இப்போ வந்து இந்த பிக்சரில் கொடுத்துருக்கு வந்து ஆவக்காய் ஊறுகாய். மாங்காயில் வந்து சால்ட்டும் போட்டு ஆயிலும் விட்டு கொஞ்சம் வெயில் வச்சோம்னாக்கா அது நல்லா ட்ரை ஆகிட்டு ஊறிக்கும் இல்லையா. ஸோ தட் இஸ் நேச்சுரல் ப்ரிசர்வேட்டிவ்ஸ். நார்மலாக வி ஆட் oil to the pickles, and the oil layer on the pickle will not allow the microbe to enter into that. So the layer of oil in the pickle will not allow the microbes to enter into that. Otherwise we say it prevents the entry of microbe. சரியா? Whereas addition of sugar, it is for jam and jelly. For jam and jelly we add sugar. So this is natural method of or natural preservatives as salt, sugar and oil. Whereas what is that synthetic preservative வந்து sodium metabisulfate. அடுத்தது பார்ப்போம். sodium metabisulfate and sodium benzoate. அது அடுத்ததுல பார்ப்போம். You just understand. Then preservation of oil and vinegar, as use of oil and vinegar prevents spoilage of pickles because bacteria cannot live in such an environment. I repeat, use of oil and vinegar prevents spoilage of pickles because bacteria cannot live in such an environment. Vegetables, fruits and fish, fish and meat are often preserved by this method. I hope you follow. So இது வந்து preservation by oil and vinegar. Then heat and cold treatment. You must observe your mother boiling milk before it is stored or used. And boiling kills many microbes. Similarly, we keep our body food in the refrigerator. So low temperature inhibits the growth of microbe. So this is heat and cold treatment. Another one is pasteurization. Pasteurized milk can be consumed without boiling as it is free from harmful microbes. The milk is heated to about 700 degrees centigrade for 15 to 30 seconds and then suddenly chilled and cooled and then stored. By doing so, it prevents the growth of microbe. This process was discovered by Viii Pasteur and it is called pasteurization. And then storage and packing, that is also a method of preservation. These days dry fruits and even vegetables are sold in sealed airtight packet to prevent the attack of microbes. The double apple which these days dry fruits and even vegetables are sold in sealed airtight packet to prevent the attack of microbes. சரியா? So இந்த ஸ்லைட் முன்னாடி வந்திருக்கணும். Synthetic preservatives, they are sodium benzoate, citric acid, vinegar, sodium metabisulfate and potassium bisulfate are added to food products like sauces, jam, jelly, packed food and ready to eat food. I repeat, sodium benzoate, citric acid, vinegar, sodium metabisulfate and potassium bisulfate. These are all synthetic preservatives. எதுலலாம் add பண்ணலாம்? Sauces, jam, jelly, packed food and ready to eat food. I hope you understand. Then these why it is added? These preservatives delay the microbial growth and keep the food safe for longer duration. சரியா? So இதில் first வந்து food preservationல நம்ம ஒரு few method பார்த்துருக்கோம். அதில் dehydration, removing the water. Dehydration is one of the oldest method of food preservation. It removes moisture from the food and so stops microbes from growing on it. இந்த கருவாடெல்லாம் காய்ச்சிடால அது. It removes moisture from the food and so stops microbes from growing on it. And dehydration also decreases the weight of the food, making it easier to store and transport. But still disadvantage வந்து some vitamins like vitamin B complex and vitamin C are destroyed when the food is dehydrated. சரியா? And then heating is a pasteurization. Heating food kills most microbes, and that's why water and milk are heated till they boil before they are drunk. Louis Pasteur found that heating food to around 60 degree, நம்ம bookல தப்பா கொடுத்திருக்கு 700 தப்பு இது. 60 degree centigrade was enough to kill most bacteria. Milk is heated to around 72 degree centigrade for about 15 seconds and rapidly cooled to around 4 degree centigrade. Correction பண்ணிக்கோ, நம்ம இதுல இருந்து 700 இருக்கு, it should be 60 to 70 degree centigrade. Okay, so this method is called pasteurization. Then refrigeration and freezing. நம்ம இது already பார்த்தாச்சு, just recollecting the points. Most microbes need optimum temperature. Most microbes need an optimum temperature to grow and multiply. Cooling food makes them inactive and also slows down their growth and multiplication. And food does not spoil for few days if it is stored at 5 degrees centigrade for months at minus 18 degree centigrade. இதெல்லாம் how far it is practically possible நமக்கு தெரியாது. இதெல்லாம் வந்து ஒரு large scale food storage-க்கு it can be followed, right? And then when the food is thawed make something warm and soft, the microbe become active again and so food has to be used up soon after. சரியா? Then vacuum packing, நம்ம bookல இல்ல, இது பார்த்துக்கோ. Vacuum packing, many microbes are aerobic. When the food is stored in vacuum, these microbes cannot grow and multiply due to lack of oxygen. This method is commonly used to store nut, grain, coffee. In the case of items like chips, which will get crushed in vacuum packed bag, the air inside is replaced with the nitrogen. You don't just a method, understand. Then canning, microbes need air and so storing food in airtight can helps to stay unspoiled for a long time. And canning, jam, fruit, vegetable and fish are used for canning method. சரியா? அடுத்தது வந்து adding preservative, நம்ம already படிச்சாச்சு. Oil and vinegar, salt, sugar, and then the chemical preservative synthetic படிச்சோம்ல, sodium benzoate, citric acid, vinegar, sodium metabisulfate, potassium bisulfate are added to food products like sauce, jam, jelly, packed food and ready to eat food. I hope you could follow this. Then next one, preservation by sugar. Jam, jelly and squashes are preserved by sugar. Sugar reduces moisture content which inhibits the growth of bacteria which spoil the food. And again heat and cold already படிச்சாச்சு நாம் படிக்கல நீ பார்த்துக்கோ. Boiling kills many microbes. Similarly, we keep our food in the refrigerator. Low temperature inhibits growth of microbes. Right? The next one and the last one, it is about the nitrogen cycle. Nitrogen cycle edukinaaka in plants nitrogen as such cannot be taken by the plants for nitrogen metabolism. It has to be taken in the form of nitrites and nitrates. So there is a there are different steps for the nitrogen cycle in which plants take up nitrogen. As first one, it is nitrogen fixation. Second one, nitrogen assimilation. Third one, it is ammonification, nitrification and denitrification. So I repeat, nitrogen fixation, it is as biological nitrogen fixers. The fixed nitrogen taking up nitrogen, that is done by mainly the bacterium, rhizobium and acetobacter, and it is always present in a root system of leguminous plant as root nodules. And they follow symbiotic association. Are you clear? And these taken up nitrogen should be entering into the cells of the roots, and this is called nitrogen assimilation. Plants take up and use nitrogen compound formed as a result of nitrogen fixation through their roots and make protein for its growth. And nitrogen thus becomes part of the plant tissues. The nitrogen is passed on to the herbivore, carnivore through the food chain and becomes part of animal tissues. Now this nitrogen compound into tissue is called nitrogen assimilation. The next one, it is ammonification. When plants and animals die, microbe decompose the dead material, decomposes fungi and bacteria, and ammonia is produced during this process. The process of formation of ammonia by fungi and bacteria, they are decomposes, which they decompose dead and waste material, is called ammonification. Next step on the nitrification. Some of the ammonia formed is taken up directly by plants, while the rest is converted into nitrates by nitrifying bacteria. And the process of converting ammonia into nitrates in the soil is called nitrification. Then some of the nitrates are absorbed by the plant. So first step on the nitrogen fixation, ammonification, and then it is into nitrification as nitrates and nitrates, and then denitrification. Right? Then as a part of nitrates in the soil is converted into gaseous nitrogen that escapes into the air. The bacteria that is involved in this process, they are called denitrifying bacteria. Example, it is what is that? Nitrosomonas, one of the species. The process of conversion of nitrates into atmospheric nitrogen, it is called denitrification. Okay, so this is for, they are further taken back to the atmosphere. So this is called nitrogen cycle with the steps as nitrogen fixation, ammonification, conversion into nitrates and nitrates, and then it is denitrification. After that, it has to be assimilated by the green plant, which is then passed on to a series of consumers in a food chain as primary consumer, secondary consumer, tertiary consumer. Again, they are decomposed. That is as a food chain where it is passed on. I hope you are able to follow this. So this video 8, it is about the microorganisms, friend and foe, and now we have dealt with the harmful activities of the bacterium and also food preservation. As due to this harmful activities, the food has to be preserved in proper way, that is food preservation. I hope you are able to follow this. So this with this what is it, microbe lesson is getting completed. So you have to go through the lesson. Apro doubt clearing session bodo Zoom session bodo you have to follow what is what, then we will continue. I hope you are following.